3. This will be a session on agricultural presentations. And just to go over a few housekeeping items, just as a reminder, this presentation will be recorded. We encourage questions, so please ask questions of the presenters. You can use the Q&A button located in your toolbar. I'm okay with if you just want to actually unmute and verbally ask. I'm okay either way with however you want to do it. So without further ado, shall we get started? Oh, and just as a reminder, you do have 10 minutes to present, but that does include your Q&A. So just be mindful of that, to leave yourself at least a minute or two for question and answers, okay? All right. Bridget, you are welcome to start sharing.
Okay, here we go. There we go. Alrighty, good afternoon, everyone. My name's Bridget, and I did my research in Dr. Carlos Rodriguez Lopez's lab. That poster will be able to download at the end of the presentation. So today I'm going to be talking to you about repurposing next generation sequencing data for more sustainable agriculture. And in this case, we are looking at NSET. And the reason we're looking at this is because in Ethiopia, NSET is a dietary starch that's high in nutrients and creates food security. So imagine being able to reduce the amount of chemicals that are being released back into the environment. And it's important to also understand that the plants act as a unit. So the microbial communities help against environmental insult and also help maintain a healthier plant. So being able to identify potential genes that signal for these microbes, you could then create a microbiome host that is able to be more sustainable in reducing non-renewable agronomic inputs that would be better for the environment and also hopefully more cost efficient. So we're looking at some of our goals was to test if genotyping by sequencing or GBS data generated to genotype crop ascensions would then be able to be repurposed to characterize the microbial communities of NSET, and to be able to characterize the leaf endobiome of the cultivated and wild type ascensions. Cultivated, another word, is also domesticated. You'll hear me use those interchangeably. So we hypothesized that the next generation sequences between wild and cultivated NSET will result in differences in the microbial communities. And to test this, we are taking published GBS data from 149 NSET cultivated and wild type ascensions of NSET to see if we can characterize microbial communities. And this right here, this graph, is the published data on the genotype of the wild and cultivated, and the genomes of the wild and domesticated are pretty significantly different. So how this was done, I was not here for this part, but it's important to understand that the endobiome is the inside of the plant's microbial communities, and that what was sampled was a leaf that was inside of the trunk because they form all the way around, creating this like dome or like onion kind of concept, where if you take the way the outside, you'd get hopefully just the endobiome of the NSET crop itself. This, in the purpose of time, or in the sake of time, I'm not going to go into detail, but if you guys would like to know more about the wet lab approach and how the it was genotyped and how the DNA was taken, you can go ahead and look at Foose paper, which is also in the poster that can be downloaded at the end of this. So here's where I come in. We took the GBS data and we did a quality check. We demultiplexed and trimmed them in to form unique sequence tags, which were then blasted against a local database where all the available genomes for bacteria and fungi were taken to determine which sequences matched up. And once we got those matchups, we went through and sorted to what I believe was a 95% match of each of the sequences, and all the ones that did not hit that match rate were then taken out. And then we looked at the abundance of these unique sequences to each of the above. So we ended up getting this data where we found that the microbial community of the wild type and domesticated showed that there was a difference. This isn't as significant as the genome one, but there is a pretty distinct line that the genome could be a driver, or the genetics could actually be a driver of the microbial communities in plants. And this graph right here shows the matchup for each of the unique, significantly unique sequence tags in retrospect or in comparison to the domesticated. So it shows that how similar or at least similar or present that they were in the domesticated NSET or cultivated. And there's a pretty strong difference down the line right here. So they're not super similar based off of this. So what we were able to conclude out of this data was that we were able to characterize the plant's microbiome using GBS data, and that also there were significant differences in the composition of the wild and cultivated NSET samples. Some further research to verify that these results are what they showed to be was in a earlier step right before we blast the unique sequence tags against the bacteria and fungi would be blasting the unique sequence tags against the NSET genome to take out any potentials that were specifically in that genome and not the bacteria and fungi. And then also to do a laboratory test of a 16S or 18S analysis, which would, since the tags were just the closest matchup, this would actually verify that these tags were those themselves. And I just want to thank Dr. Carlos Rodriguez Lopez for mentoring me, along with Lakshai Anand and Alex Stewart for helping me with the bioinformatic analysis, and also the Student Sustainability Council and undergraduate research for supporting the fellowship. If you guys would like, there's the QR code to download the poster. And if you guys have any further questions, you can contact me at that email. But thank you guys for listening today. Do you guys have any questions? I don't know.
I think I have a question. I don't know if you already explained this, but when you were talking about like the onion shape, so like the inner layers would probably be the newest, right? Like the youngest leaves or whatever.
Yeah, and they'd have no exposure to the outside microbiome or like the microbial community around there too. So the environmental factor would just be looking at that.
Okay. So then like the microbiome that would be in the outside layer would be different than the one on the inside because the outside is like interacting with the environment? Okay.
Hypothetically, yes. I don't have proof of that, but it should be because you have air, animals, humans walking around on the outside, so, or insects too. So it should have other factors that play into it.
Okay, thanks.
No problem, thank you. I might have to stop sharing my screen to see if there's any questions in the chat, but I'm not sure. Oh, there is, I see. How do wild and domesticated insects differ? So wild and domesticated, one is the one that's kind of bred for eating, which got rid of a lot of the extra factors of like their berries and fruits. It focused more on the actual edible part that they make is the crom or corm, which they ferment, and that's the nutritious part. Whereas the wild type, it's not as edible or what they use for eating. So it takes away the factors that aren't used with the domesticated. Yeah. I think that hopefully that answered your question.
So I have a question, Bridget.
Mhm.
What impact or what hope do you have this research will actually have in Ethiopia?
Well, it's partially in Ethiopia, but also just in general, being able to identify different gene markers that control and like not even just NSAID's microbiome or endobiome, sorry, but other crops, you'd be able to kind of make it easier for crops to produce what they need to and more efficient, and also hopefully making it a little bit more environmentally friendly.
Gotcha.
But
So that it would serve across populations, not just specifically Ethiopia.
yeah,
Okay.
and yeah, because for this project, the reason that it's kind of interesting is I took out lab work of using additive plastic, of doing extra samples or running samples. I took previously run samples and did a bioinformatic analysis to see if it could be kind of repurposed for something else, or to see with the microbiome instead of just the genome.
Interesting.
Yeah. Well, thank you all for listening today.
Absolutely. Nice presentation, Bridget.
Thank you.
All right. Our next presenter is going to be Rebecca Caldbeck. Did I pronounce that right?
Yeah, thank you. Yes, ma'am.
Okay, good. Yay. Are you ready to start?
Ma'am?
Okay.
Can everybody see that?
There we go. Put it in presentation mode. Yep, there you go. Perfect.
So hello everyone, my name is Rebecca and I'm a senior that's studying Ag Biotech and Environmental Science, and today I'm going to be presenting on my project regarding understanding the feasibility of using like hemoglobin protein as a cisgenic marker for crop improvement. So the global population is expanding rapidly, as we all know, and UN estimates say that we will likely reach almost 10 billion people by 2050. That's a lot of mouths to feed, so we will definitely be putting an even larger strain on food production, which will lead to further decline of ecosystem health. So it's now more important than ever to focus our efforts towards sustainable crop improvement. The preferred route for this improvement would be cisgenic modification. This is mainly due to the lack of cumbersome regulation surrounding it. Cisgenic modification involves a recipient and donor from crossable species, while the more regulation-heavy transgenic method of modification involves a recipient and donor from non-crossable species, such as using genes of bacterial origin in a plant. To make these genetic improvements, it's essential to have genetic markers. In this study, I focused on testing a marker that I extracted from glycine max, which is commonly known as soybean. Soybeans are a legume, which means they are capable of fixing their own nitrogen thanks to a symbiotic relationship in their roots. One protein that's contained in the root nodules is like hemoglobin. It's related to myoglobin and hemoglobin that can be found in animals, and it contributes to the red coloration that can be seen in nodules, as shown in figure 2 up in the top right. So the primary question behind this project was to analyze the feasibility of using this like hemoglobin protein as a cisgenic visual marker for soybeans. In order to do this, the goal was to create transformants that express like hemoglobin's red pigment. The red pigment would theoretically be useful in screening and detecting genetic improvements more efficient. Before using the marker in soybeans, we studied Arabidopsis. This is a model species used in plant research, and it's very space efficient and fast growing, which helps speed up the research process. If the marker is successful, we will then transition to soybeans. In addition to soybeans specifically, having an effective visual marker to facilitate genetic improvement would be useful for further development of more nutritional food products in general for a variety of crops. So if successful, this project has the potential to have impacts on the food, health, and agricultural industries. To begin the project, like hemoglobin primers were first developed. The RNA was extracted from the root nodules of the soybeans, shown in Figure 2. Red coloration indicates a healthy nodule, so special care was taken in sample collection. The extracted RNA was used to synthesize cDNA. The cDNA was then amplified using PCR with the specifically designed primers and validated using agarose gel analysis. Overnight cultures were prepared and then mini prep was performed to purify. A restriction enzyme digestion was performed and verified again using the agarose gel extraction analysis. The PCR product and the vector, shown here in Figure 4, underwent ligation. The ligation results were transformed into E. coli bacterial cells using heat shock and then plated to grow the colonies. So like hemoglobin protein was successfully extracted from the nodules and then amplified using the designed PCR primers. Like hemoglobin has a known band length of 464 base pairs. If you look down there at Figure 6, you can see that nodule samples 1 and 2 both contained a like hemoglobin band around this known 464 base pair value. The vector and amplified products were digested using restriction enzymes and displayed the expected results, indicating linearization of the vector, as seen in Figure 5. Figure 1 displays the process of linearization using restriction enzymes. Restriction enzyme digestion is basically using enzymes like hypothetical scissors to cut the backbone in a specific place. So basically the vector began as a circle, and once the enzyme scissors were used, it became linearized. Once both components were linear, the vector and the nodule product could be ligated. Ligation refers to essentially gluing the pieces together with the help of the ligase enzyme, and it's described in the visual in Figure 3. So you start with 2 sticky ends, and then they're stuck together using the ligase enzyme, and it forms a recombinant DNA. The ligated product underwent transformation using heat shock to stun the E. coli cells, which then allowed the desired product to enter and begin production. The product was spread on a plate, and I'm now waiting for colonies to grow before proceeding. Due to COVID-related delays, no substantial conclusion on feasibility can be made yet, but the experiment so far has produced expected results in terms of gel results. Overall, it's been a fantastic experience, and I hope to continue the research in the future. And I would like to acknowledge my PI, Dr. Kasheru, and all others who have aided in my academic efforts, specifically also Dr. Hildebrandt. Thank you all for listening. Do you have any questions for me?
Nice presentation, Rebecca. I like your poster.
Thanks.
It's visually stimulating. What were some of the challenges that you faced doing this research?
So due to COVID, it was definitely a struggle getting started at the beginning, just due to the restrictions and getting started back in the lab again. So that kind of held up the experiment. And then also getting the primers designed was a challenge that I had to overcome, but eventually got those out and they worked. So that's good, but it did slow down the process as well.
Dr. Mo has a question for you in the chat, and it says I cannot say it. Yeah, is natively present in soybean roots, right? How can you tell the native from transformed plants?
How can I call a native from transformed plants?
How can you tell native from transformed plants?
Okay, so the like hemoglobin protein is pink in color, and our hypothesis is that the like hemoglobin protein will be expressed as a pink color when attached to a gene of interest in a transformant. I hope that answered the question.
Okay.
Any more questions?
So I just have one, Rebecca. So what does your future look like? Are you going to continue this research, or what year are you? Are you graduating?
Yes, ma'am. So I'm graduating and I'm majoring in ag and medical biotechnology and environmental science. So this project was like the perfect combination of my two focuses, and using biotechnology to develop these markers that can assist in genetic improvement, which we know is essential for making food production more sustainable and environmentally friendly. But plans for finishing the project, ideally, if it wasn't for the COVID restrictions, I would already have an answer to the feasibility, but although I didn't get to fully complete the work just yet, I do plan to continue the work for the next several weeks before the summer and hopefully reach a conclusion then. But the project has been a wonderful learning experience despite the setbacks and challenges.
Sure.
Solidified my love for research.
That's awesome. Awesome. All right. Any other questions for Rebecca before we move on? All right. Our next presenter then is Anna Foose. Anna, are you ready to present?
Yes, I am.
All right. You may share your screen.
Okay. I apologize for any background sound. There's people playing music outside, so I really can't control the sound right now. Okay.
The good news is I can't hear it.
Yeah.
So.
Okay, so my name is Anna Foose, and today I'm going to be talking about the role of grooming behavior in pesticide resistance and infection in honeybees. And my research mentor is Dr. Claire Richoff in the Department of Entomology. So before we get started, there are two kind of important definitions to know. The first is that self-grooming is when a honeybee uses its legs to kind of like wipe its body off and get any dirt or debris off of its body. And then allogrooming is when another bee comes up and basically grooms another bee. So it's a type of social interaction. Previous research in the Richoff lab found that injecting honeybees with yeast cells actually induces this allogrooming social interaction, and that colony members are able to target their allogrooming to those diseased individuals. So the ones that were injected with yeast cells were expressing some type of like distress, and the other bees were able to come up and groom that individual in an attempt to like clear the yeast infection, which of course this doesn't make a lot of sense when a yeast infection is internal. So grooming the bee from the outside isn't really going to help clear the yeast, but somehow the bees were still able to like target these grooming efforts to the sick bee. So in this study, we kind of expanded upon that and evaluated how allogrooming and self-grooming benefits the infected bee, and then how that benefits the colony as a whole. So my hypothesis was that receiving grooming will increase survival when individuals are challenged with either yeast or an additional stressor of a pesticide. And the pesticide that we used was tetradiphon, which is a very common pesticide used in agricultural fields where honeybees would be foraging. And so to test that hypothesis, we compared the survival rates between individuals who received that allogrooming and then individuals that were placed in an isolation setting where they weren't receiving any allogrooming at all. And then we looked at the combination of yeast treatment and then yeast and pesticide interactions. So moving on to methods, there were a total of 6 different treatment groups, and then we had 2 different experiments to treat all of, or to test all of those treatment groups because there were a lot. So the first experiment, we treated 2 bees with a yeast from each of the 10 hives that we were investigating, and then placed them in either a social or isolation setting. And then in the second experiment, we treated 2 bees with either yeast and a pesticide or no pesticide, and then placed them in either social or isolation settings. In this picture here, you can see an example of what the social setting would look like. So this bee, it's very small, so it might be hard to see, but this bee has a little red dot of paint. So that was our treated bee that we were paying attention to. And then these 3 bees are from the same hive, but weren't treated with anything, and they were only there for the purpose of providing grooming for this treated bee. And we observed these bees over the course of an hour and marked how often they self-groomed if they were on their own, and then how many times they received the owl grooming if they were in this social setting. So we had some interesting results. Here in this first graph, we have the social setting and how many times the bee self-groomed on average per minute. So we can see here in gray and in yellow, these are the pesticide and yeast and pesticide treated bees. And so both of these groups had higher rates of grooming than the bees that received no treatment at all, and then bees that were treated with a yeast. And in this bottom graph, we have the isolation setting, so where the bee was on its own. And the yeast treated bees actually self-groomed less than the untreated bees, which was a very surprising result, and we weren't expecting that at all. And the pesticide treated bees, on the other hand, had the highest rate of average or self-grooming rate per minute, and the combination of yeast and pesticide treatments lowered that average rate of grooming to about 1 time per minute. Let me move these videos. So here in the results in this graph, or sorry, let me rewind really quick. So overall, we found that owl grooming was a very poor predictor of survival after 24 hours, but we did see connections between the amount of average self-grooming and saw that that was a pretty good predictor of if the bee was going to be alive after 24 hours. So in this graph on this side, we have if the focal bee, so the treated bee, if it was alive after 24 hours, and then either yes or no. And on this side, we have the social setting, so no for isolation and yes for the social setting. And then down here, we have the average rate of self-grooming. So we can see here in the yes row for bees that were alive after 24 hours, they had the highest levels of average self-grooming per minute, and the bees that died within the 24 hours had lower rates of self-grooming. So from this, we concluded that self-grooming was a good predictor that the bee was going to survive the treatment of the yeast and the pesticide. We had some limitations in this experiment. So overall, we saw an 88% survival rate while we were expecting about a 10% survival rate. So that kind of made me think maybe our yeast culture wasn't quite right, or our application of the pesticide in the yeast to the bees wasn't effective, since so many survived, and we predicted that about only 10% would survive based on previous research. And there's many applications to this research. The biggest takeaway was that beekeepers may want to favor aggressive breeds of honeybees, because previous research has found that aggressive bees are actually more likely to groom in general. So if they have higher rates of this self-grooming and owl grooming, then they're more likely to survive stressors that they might encounter while they're flying around in agricultural fields where there's yeast and all kinds of pesticides sprayed on the flowers that they might become infected with. And I wanted to say thank you to Dr. Richoff, who's been my mentor for the past 2 years, and then PhD student Rebecca Westwick, who helped me with this entire experiment, and Jimmy Harrison, who was our lab technician during this experiment. I'll take any questions that you all might have.
I don't know if you've specified this, I might have missed it, but what's the importance of bee grooming?
So bees will kind of like their main goal is to maintain cleanliness in the hive. So they'll want to clear any like dirt that might have gotten stuck to them while they were out pollinating flowers. So they don't want a bunch of buildup of debris in the hive. So they kind of clean each other off and then try to remove like any varroa mites are a very common pest, and they're basically just like little bugs that attach to the bee. So they'll try to like scrape that off to prevent infestation in the hive.
Oh, okay, thank you.
Oh, I think there's a human.
And there is a question in the chat by Luke or Dr. Mo. He said, how did you isolate the yeast, and is this a pathogen for bees?
I believe it is a common pathogen for bees, and we actually had to, we cultured it in a liquid and then injected it into the bees. So the bees were anesthetized on ice, so they were asleep, and then we injected them with the yeast culture. And then when they woke up, they were infected with the yeast. And this is commonly done in other experiments in the Richoff lab, and in other labs, it's typical to artificially induce this yeast infection through injection.
Anna, what do you hope to do with this research, if anything?
Well, in general, I'm a huge advocate for insects and honeybees and pollinators in general. So I think hopefully this increases some of the conservation efforts for honeybees, and in general, any knowledge we have about our pollinators is going to be helpful for conservation in the future.
Nice, thank you.
Thanks.
Does anybody have any other questions before we move on for Anna? All right, well, very nice presentation, Anna. All right, our next presenter is going to be Madison Sydnor.
Yeah.
Sydnor. Okay. Are you ready?
Yeah, I'm
Madison?
ready.
Okay,
Thank you.
sure.
Okay, can you all see this?
Not
and
yet.
Hear me okay?
There we go.
Okay. I think my Wi-Fi is a little slow today, so if you ever need me to like pause or repeat something, let me know. Okay, so my name is Madison Sydnor. I'm a senior studying biology and environmental and sustainability studies, and I worked with Travis Bennett, a graduate student mentor on this project, and Dr. Hannah Poffenbarger, my faculty mentor. We examined how chemical dispersants, varying land use, and soil depth impact our recovery of soil organic matter. Healthy soil, as we all know, is the foundation to sustainable societies and the solving of many issues tied to climate change. Here are just 3 of the United Nations Sustainable Development Goals, which are directly reliant upon healthy soils. So first, goal number 2, healthy soil is required to maximize our food growing capacity to meet the growing hunger demand. Goal number 13, soil is a really important carbon sink and is home to many global and regional geochemical and nutrient cycles. And also, healthy soil is the foundation to life on land and creating environments which are resilient and protected against degradatory processes like desertification. Soil organic matter, or SOM, is key to healthy and productive soils. It affects all soil properties such as water holding capacity, cation exchange, decomposition, and many others, which are shown here in this figure. And overall, it is essential for healthy plants. SOM is incredibly complex and requires separation into multiple parts in a lab setting. And this can be problematic because there are many methods of separation. This table shows one study which examined 20 different methods, but this list is not even an exhaustive list. So there's a need in the field to standardize these separation methods. So recently, a framework for separation that has gained popularity suggests that SOM can be separated into particulate and mineral associated organic matter. And this is because these fractions differ based on properties like formation, persistence, and function in the soil. Particulate organic matter, or POM, is the less protected fragment and is more available to be decomposed. It's basically like plant matter, little twigs and leaf litter that hasn't been decomposed yet.
And the mineral associated organic matter, or MALM fragment, is more stable and is protected from decomposition. It's also very rich in carbon and is found in the soil, associating through adsorption to rock and mineral surfaces. So these fragments are often dispersed using chemical dispersants, but these chemical dispersants can distort how the carbon is arranged in these fractions and prevents us from depicting a complete picture of what SOM looks like in our natural environment. So this leads us to our research questions, which ask, 1, can DI water, distilled water, be used for separation as an alternative to chemical dispersants? And specifically, we looked at sodium hexametaphosphate, which is a very commonly used chemical dispersant. And second, is DI water effective for dispersing soils across differing environmental conditions? And specifically, we looked at differing land uses and soil depths. And so to test these questions, we first collected surface and subsoil samples from corn and sod plots. We then dispersed the soil with sodium hexametaphosphate and DI. Then we separated them into their POM and MALM fragments using a 53 micrometer sieve. And then finally, we collected the masses of the dry soil. And so here are results which answer our first question. We hypothesized that compared to the commonly used sodium hexametaphosphate, DI water would comparably disperse soils. And again, this is important because chemical dispersants can distort and mask the arrangement of the organic matter in our samples. So the top figure shows us that the percent recovery from each dispersant. You can see DI on the left, sodium hexametaphosphate on the right, percent recovery. And from this graph, we concluded that DI water was an effective dispersant. And additionally, we see that sodium hexametaphosphate recovery was a little above 100%, around 101, 102. And this is because sodium hexametaphosphate holds on to water and thus accounts for this greater than 100% recovery. And this is another reason why we think DI water may be preferred to sodium hexametaphosphate in certain situations. And additionally, as shown in the bottom figure here, both DI and sodium hexametaphosphate result in equal recovery of each fraction. So here is our particulate organic matter recovery, and there are no statistically significant differences between these two recoveries. And this is our mineral associated organic matter recovery. There's also no statistically significant differences here on recovery. And once we confirmed that DI water is a suitable dispersant, we wanted to examine its efficacy across environmental conditions. And we hypothesized that DI water would still be an effective dispersant across environmental conditions, and specifically land use and soil depth. So in this figure, we have the recovery efficiency graphed for our two land use conditions, corn and sod, and there were no statistically significant differences between these two recoveries. And in this next figure, we have the recovery efficiency for our three soil depths. So this is our surface soil, and these are our subsurface soils. And there were no statistically significant differences between these three recoveries. And from these two figures here, we concluded that DI water is a suitable dispersant across varying environmental conditions. And to summarize, both of our hypotheses were supported. First, we concluded that DI water was an effective alternative dispersant to sodium hexametaphosphate and may be preferred because it doesn't distort the carbon arrangement in our samples and doesn't cling to our samples, causing recoveries over 100%. And second, we concluded that DI water could be widely applied as a dispersant across environmental conditions. And these findings are interesting because this framework and dispersion using DI water is cheaper, accessible, it's equally effective to chemical dispersants while preserving carbon integrity of our samples. And also it's a really dynamic framework and can be widely applied to soils across varying environmental conditions such as land use, different soil depths. So it's a really accessible and widely applied framework. And also, it's important to have methods to evaluate the natural state of organic matter without disrupting carbon, so we can better understand how soil organic matter develops and changes over time. And knowing this, we can better advise strategies to manage land so it retains carbon over time, and so we can rebuild carbon in soils and maximize soil richness. And in doing so, we can better support progress towards these specific targets of the UN Sustainable Development Goals and sustainable agriculture as a whole. So those are all my slides. If you guys have any questions at this time, I'd be happy to answer them. And I can go back to any slide too.
What got you interested in soil science?
So I'm working currently as an undergraduate assistant in an agroecosystem and nutrient cycling lab. And not that I came into the lab with like a huge interest in soil specifically, but I want to go into a career in sustainability. So I felt like it was like a way to incorporate sustainability and more of like the hard technical sciences, since I do have a background in biology. And also just because the opportunity presented itself to start working there over the winter. And I ended up really liking the people I work with and liking this project a lot.
Very nice, Madison.
Thank you. Are there any questions in the chat maybe? Let me stop sharing.
Is this for
It looks like maybe the error bars were greater for the DI water. Is this a concern? From like our discussions of the data, it didn't come up in our conversations and didn't really seem like a concern to us. I think that it's more just natural variability in the data. But yeah, does that answer your question?
All right. Very nicely done, Madison.
Thank you.
One last opportunity to ask Madison any questions before we move on.
There's one in the chat that says, do you think POM or MAOM is better for soil health? From Hannah.
I think it's hard to say. So they both serve very different functions in the soil. The palm is more, I guess, short-lived and has not been decomposed by microorganisms yet. Whereas MAOM is the organic matter that sticks around in the soil for a long time. So if you're thinking like about soil resilience and like development of these carbon sinks for like the long term, I think MAOM is the fraction that better serves that purpose. But yeah, they both serve really different functions in the soil. So I think it's more on like a case by case basis, which one is more important for soil health.
All right. Well, again, thank you, Madison. Great job. Okay, moving to our next panelist will be Sabria Orion. Did I pronounce that right, Sabria?
Sabria Arain.
Sabria Arain. Sorry about that, Sabria.
Fine.
All right, are you ready to start?
Yes, I'll share my screen.
Okay.
Okay, can everyone see it well?
Yes.
Okay, perfect. So my name is Sabriyah Arain. I'm a senior this year, and I'll be graduating with a degree in marketing and merchandising apparel and textiles. And my research was about a global pandemic's effect on the retail industry, and my mentor was Dr. Scarlett Wesley. So the COVID-19 pandemic has changed the retail industry since it first began. Consumers are now choosing to stay safe at home rather than go out to stores to shop. And so retailers are facing consumer demand and marketing issues as a result of this trend. It's anticipated that the retail industry won't look the same after the pandemic subsides, so companies need to make sure to change their operations in order to survive and continue to be successful. So my overall research goal was to discover how COVID-19 has impacted the retail industry by evaluating changes that occurred during the 2020 holiday season as compared to the 2019 holiday season. I hypothesized that the retail industry would likely experience shifts in operations that would continue after the pandemic is over. And to meet my research goal, I chose to analyze 3 distinct categories, which included visual displays, shopping patterns, and marketing tactics. So to analyze visual displays, I chose to look at Instagram posts from 2019 and 2020 during the holiday season, which is roughly from November 1st to December 31st. I chose to analyze 4 different retailers, which were Altered State, Express, Calypso Boutique, and Ely's Contemporary Boutique. All of these retailers have similar product lines and similar target markets, so I thought that it would be good to compare them. And I chose to analyze the 2 mall stores, which were Altered State and Express, because I wanted to see how the pandemic has affected the retail industry on a more national level. And then I chose Calypso Boutique and Ely's Boutique because I wanted to see how the pandemic affected retailers on a more local level as well. And then to analyze shopping patterns, I conducted a survey to UK students regarding changes in consumption patterns during the 2019 and 2020 seasons. I received a total of 56 respondents from this survey. And then lastly, to look at marketing tactics, I interviewed the managers from the 4 retailers previously mentioned, and we discussed changes in marketing strategies between the 2 holiday seasons. So the first thing I analyzed was the visual displays for the 4 retailers, and I looked at their Instagram posts from both 2019 and 2020 during the holiday season. And so this chart just kind of shows a summary of the data I found. So you can see that the average number of weekly posts changed between 2019 and 2020, and the content changed. They advertised completely different products between the 2 seasons as well. And so these are the main patterns I found based on that data. Other than Altered State, all retailers increased their number of weekly posts. And this is likely due to the fact that since we were in a pandemic and many countries were on lockdown, like in the US was on lockdown for quite some time, and like lots of stores were closed. So this meant that more consumers were spending time on social media and shopping online. So these retailers wanted to take advantage of this trend and continue to post so that way they would still remain relevant during this time. And then also the content of posts change. For all 4 retailers, there was an introduction of loungewear and more comfortable and casual clothing. And this was also likely due to the trend that since more people were spending time indoors, they needed to create products that fit this lifestyle. And then lastly, there was an increase in promotional sales. And this was likely due to the fact that since not a lot of consumers were entering stores anymore, retailers still wanted to encourage them to continue to shop and remain loyal to their companies. And so next is the shopping patterns analysis. And so from the survey I conducted to UK students, I came up with 3 major points that were very important. So first, 78.57% of respondents made more online gift purchases this past holiday season, and many attributed this to the safety and convenience of online shopping rather than going shopping in store during a pandemic. And then also 67.86% of respondents still purchased the same number of gifts in 2020 because they still had to shop for the same number of individuals. And these individuals mainly included family, friends, and siblings. And so this data just shows how quickly consumers can respond to changes in the retail industry itself. And then lastly, 55.36% of respondents noticed more promotional advertising during the 2020 holiday season than the 2019 season. And so lastly is the marketing tactics analysis. These are the major changes I saw between 2019 and 2020 based on the interviews from the 4 retailers. So besides Calypso Boutique, all retailers experienced at least a 20% decrease in foot traffic compared to the 2019 season. There were also changes in marketing tactics. Retailers used heavier social media, more convenient pickup options, and they chose to reach out to customers through online methods rather than in-store methods or mail methods. And then there were new product lines. Each retailer decided to create loungewear and other comfortable clothing lines to further relate to the current lifestyle that many consumers were taking part in. And then besides Calypso Boutique, all retailers changed their in-store signage to appeal more to customers. So this included simplified window displays and posting more frequent in-store signage to advertise sales. And then personalized promotional events were also increased because these retailers wanted to further encourage customers to come in-store to shop. And so in conclusion, it can be hypothesized that the retail industry will not return back to normal once the pandemic subsides. And there are three major changes that can be predicted. First, social media marketing will continue to be a major part of retail operations. Social media content will continue to be more creative, and digital strategies will become a primary focus in the retail industry. And then next, there will be an increase in online shopping. Consumers will start to get used to the ease and convenience of using these methods and start to view in-store shopping as less and less necessary. And then last, there will be a change in product lines. There will be a rise in loungewear and casual clothing that began during the pandemic, and there will likely be a new trend of home wardrobes that many retailers will adopt. And that is the end of my presentation. Thank you all for listening, and I'd be happy to answer any questions you have.
Did you find any results that you weren't expecting?
One result that I wasn't expecting is that Calypso Boutique in particular did not experience a lot of changes in foot traffic and didn't really change their marketing methods either. And when I was speaking to the manager, they attributed this mostly to the fact that many of their consumers are local and already know what they sell, so they didn't really have to try to get any new customers since their base is already so solid. So that shocked me that it was kind of a difference between local and national retailers, and that local retailers almost had less things to worry about just because they were already so well known in the area. Part of me, so the question is, do you think the increase in social media would have happened without the pandemic? And I believe that it would have still increased, but not to such a large extent, because due to the pandemic, many people were on their phones more and were using social media more often. And so I think that many retailers took advantage of this trend and decided to advertise more in social media, TikTok, Instagram, and they chose to further push their products onto these platforms because they knew that these consumers were using them even more during the times when they were spending more time at home.
Nice job. Very nice presentation. Very relevant, huh?
Yes.
And I agree. I think we're going to see a lot of things that retailers have adapted to COVID that they will continue. And especially, I think, the small business. Not necessarily the larger corporations, but I think the small business owner will continue to do more online, or at least hire social media folks who can do this for their marketing for them.
Yes, I think that it's made a lot of local retailers realize that they have the potential to reach a larger audience through these social media methods as well.
Yeah. Awesome. So what are your plans, Sabriyah? Are you a senior, or are you coming, you still another year?
I am a senior. So as far as this research study goes, I don't think I'll be doing too much work with it, but I know I'll continue just to analyze trends in the retail industry itself. I'm actually going to be working as a digital marketing coordinator after graduation. So it's very relevant because, as I mentioned in my presentation, like, digital marketing is going to become increasingly important. So I'll be glad to kind of apply the research that I gained from this project.
Absolutely. Well, awesome. Well, great job presenting. And good luck in your new position.
Thank you.
All right. So now we are going to switch to kinesiology and health promotions. And our next presenter is going to be Julia Estes and Camille Wright. Are you both presenting?
Yes, so I'll be sharing the PowerPoint and we'll just be switching off with
Okay,
each slide.
sounds good, Camille.
No problem. Let me continue sharing.
Oh, there you go. Let me see. Did it?
Is
Yeah, it
everyone
stopped.
able to see that?
No, it's there you go.
Okay, cool. So I'll kick us off now. So my name is Camille Wright.
And I'm Julia Estes.
And we've been conducting research with hashtag I Can End the Trend, which is a peer-led e-cigarette program here at UK. Our research, we decided to connect with Markey Cancer Center to conduct this research, and we're conducting it with the MCC Appalachian Career Training in Oncology program, also known as ACTION program. We'd like to thank our research mentor, Dr. Ickes, and program coordinator, Sierra Miley, for assisting us with our research this semester. And for our research, we evaluated our peer-led e-cigarette program, hashtag I Can End the Trend, and the effectiveness in our intervention with Appalachian youth.
So I Can End the Trend was developed by Breathe faculty at University of Kentucky in collaboration with Tobacco-Free Take Action ambassadors. The primary goal of our program is to provide tobacco use prevention services to youth, families, and community members across Kentucky. I Can End the Trend is an evidence-informed program to help build awareness on the dangers of tobacco use, including e-cigarette use, cost associated with nicotine dependence, and ways youth have been targeted and influenced to use these products. Our team strives to empower younger generations to end the trend of e-cigarette use among their peers and in their communities through skill-based and engaging lessons. We design the lessons based on national and state education standards, as well as feedback and lessons learned from previous presentations, which we will share with you guys today.
So hashtag I Can End the Trend partnered with the ACTION program in order to deliver a two-part presentation to high school students in the program. So ACTION, or Appalachian Career Training in Oncology, offers Appalachian Kentucky high school and UK undergrads opportunities for cancer research, clinical outreach, and education experiences. So studies have shown that young people in Appalachia especially have higher tobacco rates than other areas of the US, and that young people in Appalachia, they're less likely to think that e-cigarettes are harmful. So our goals of the partnership were to reach Appalachian students in order to educate them about e-cigarettes and encourage them to participate in advocacy engagement activities to decrease those e-cig rates.
Part one of the two-part presentation consisted of education regarding the history of tobacco, the health and financial consequences of using tobacco and e-cigarettes, how the tobacco industry uses manipulation and targeted advertising to reach young people and Appalachian youth, and what youth advocacy looks like. We also had discussion activities and Kahoots. Part two consisted of reviewing that tobacco education from lesson one, discussing why these youth want to be advocates and the barriers that can come with advocacy. And we also included interactive advocacy activities and again, discussion activities and Kahoots.
So in terms of our participant data, all of our participants, they were all upperclassmen in Appalachian high schools. They were either 14 or 15 years old. The majority were white and all were non-Hispanic. It was mainly ninth and 10th graders. There were a little bit more female students at our sessions than male students, but there was still a very good balance. And yeah, and Julia's gonna go into more detail about this data here.
So participants were sent a link to an online survey through Qualtrics, which is a secure survey database following the training. The post-survey aimed to evaluate the effectiveness of the partnership, how much the participants learned, and how they responded to the virtual platform. So all students reported that they learned something new about e-cigarettes. They felt confident to be an advocate in their school and community, that they enjoyed having a college student facilitator present the program, and that the facilitator kept them engaged virtually through hands-on activities and opportunities. We did run into some barriers though. So when asked what participants would like to change about the program, common answers included the length of the presentation. So the first part of the session was around 7 hours long, and the second part was much shorter, around 3 to 4 hours. So a lot of the participants experienced Zoom fatigue, which I'm sure we're all very familiar with. Participants were very tired after the first session and voiced a desire for a shorter session in the future. They also wanted our presentations to be faster and less repetitive. Another barrier is the perceived tobacco culture of the state of Kentucky. So many young people have seen their older relatives and family members use tobacco products their entire lives, and it makes it difficult for these young people to even imagine the end of tobacco use, and it can be a little discouraging.
So some lessons that we learned or that rural teens, they were engaged and empowered after program participation, even though it was very long and they were definitely tired, they were definitely still stayed engaged. And that was definitely due to the virtual games that we kept doing and asking them questions and just keeping them feeling present throughout the day during those sessions. Also, youth, they definitely do have a desire to be involved in advocacy efforts. It's just a lot of them, they don't know about these efforts. So in the sessions, we were making sure to highlight lots of opportunities for them. And a lot of them did end up signing up for most of them, if not all of those opportunities that we did list, and would reach out to us via social media or emails, letting us know that we participated in this, we loved it, and they really appreciated us giving them more ways for them to be more active with this issue. And like I said, Zoom fatigue was definitely a problem, but doing those activities like Kahoot, different scenario activities, those were really great ways in keeping the youth super engaged, even though it was a very long session. So moving forward, we've realized that using not necessarily a college aid facilitator, but a facilitator that is close in age to the students that they are presenting to is a very effective way to communicate with students because it allows them to feel just way more comfortable and honest during the program, and it can be more candid with you. And lastly, there's definitely a need for additional tailored presentations so that we're able to reach youth that are in different communities like Appalachia and all over Kentucky. So are there any questions?
Hi, ladies. Great job presenting. I love the way you tag teamed. I have a question. Were your high school students, did they participate? Like, were they smokers or were they e-cigarette smokers or did they chew tobacco? So were they participants already of this? Or do you know?
We didn't know if they used before this program. We couldn't collect that data, but we did provide quitting resources and quitting tips in case they were users. and ways that they can help their friends that use e-cigarettes or tobacco quit. But we did not know if they use tobacco or e-cigarettes.
Gotcha. Any other questions?
Is there anything that you guys found out of doing this? Like, what methods do you think you guys should have tried with it, or like for future? Like, I know you guys said like shorter lessons and like someone closer to their age, but was there anything that you found like shocking, or not shocking, but interesting, that clicked that you didn't think would?
I was honestly surprised that as many students I was surprised that so many students were engaged throughout the 7-hour presentation on a Saturday. So even though that was our biggest, takeaway, that we should make it shorter, it still wasn't an overwhelming amount of people saying that, the length ruined the presentation. It was more of just an additional suggestion. So I was honestly surprised how willing these high schoolers were to set aside a whole 7-hour day on a Saturday just to learn about e-cigarette prevention.
Something I was surprised about was, well, at the end of the whole program, the students, they have to practice and record themselves like giving an actual e-cigarette presentation like they would give to other students or just their peers or something. And a lot of them were super, super good at it. And like that I was definitely their strength presenting and like giving information. And they're also super passionate about not just even e-cigs, but they all had like a very awesome variety of different passions as well. We tried our best to, whenever we were doing any sort of advocacy like training and like having them practice writing a statement to send to like a legislator or something, we wanted them to feel like they don't necessarily have to make the statement like about e-cigarettes, but whatever they're passionate about, just like go for it and try to make that statement. And they were all just like so good at it and have so many things that they're so incredibly passionate about. I think as they like keep stepping more into these roles and like when they get older and come to college, they're going to be like so ahead of the game. I was really pleasantly surprised by that.
That's
How were
fun.
these students recruited? Like, did you recruit them through the high school itself? And were there any incentives for them to do this, do you think?
So they were participants in the action program through UK. And
Oh, okay.
yeah, so they already had an interest in more so cancer research and opportunities through that. And then the leaders of action presented the students with this opportunity.
Okay. You said that in the beginning and sorry. Yeah.
You're fine.
Yeah, because 7 days on a Saturday for a 14-year-old, that's quite impressive.
Yes, very impressive.
All right. Does anyone else have any questions for Camille or Julia? Great presentation, ladies.
Thank you.
So, all right. Well, that concludes our session for today. Thank you all very much for participating. And it was wonderful to hear the variety of presentations. You all do such wonderful things here. I'm always amazed. So I hope you have a good rest of your semester. What, you have 1 week left, right? And wherever your next journey is, I hope it suits you well.
Thank you.
Thank you. Bye.
Thank you.
Bye.